Je vous remercie. Je voudrais tout d'abord remercier les organisateurs, Edith, Geneviève, Monsieur Cusin et Monsieur Bach, de m'avoir invité à cette conférence qui est vraiment fantastique. C'est un moment un peu exceptionnel, je trouve, parce que les académies, normalement, sont des institutions relativement traditionnelles. Et là, depuis ce matin, on est en train de parler d'un sujet vraiment non conventionnel, quasiment antidogmatique, dans un endroit absolument prestigieux, sous les auspices de l'Académie, devant une audience... je suis désolée, je viens de faire ça en français sans le réaliser, devant une audience exceptionnelle également.
En français, c'était agréable à écouter aussi, vous pouvez passer un...
I was saying, for my non-French, I was saying that it's quite an exceptional moment because usually academies are quite traditional, and here we've been talking about something non-traditional at all, quite unconventional, even anti-dogmatic, in this very prestigious place, and with the care and the auspices of the Académie. So to me, at least, I think it's quite a memorable moment. I had not thought I would live this once in my life, so I think it's really a great step forward for the field. Who knows? Monsieur Bach this morning said that when you have to prove such a very difficult concept, you better bring very good data. So I'll do my best to show you the most convincing data, and I hope I will convince you that epigenetic inheritance in mice is true. So I will particularly do so in the context of behavior and traumatic stress. So this is just a generic slide to show that we are influenced by many different factors, whether food, nutrition, we've heard about this, drug, chemicals, stress, pollution, environment, but also lifestyle and exercise, and that potentially all our cells can be modified. And our cells can be modified really throughout life, from the very early on to throughout embryogenesis and postnatal development, and even in old age, although, like de Lamarck, I believe that adult life and aging is less sensitive. When we talk about epigenetic transgenerational inheritance or germ cells, that germ cells are less sensitive to the environment in grown or old age or adult stages. But indeed, embryogenesis and early postnatal life are really thought, and I think there is good evidence now to confirm this, to be the most sensitive periods. And I'm a neurobiologist who has always been interested in psychiatry, there is a very intriguing problem in psychiatry, is that there are many diseases like depression, schizophrenia, borderline personality disorder, who were thought to be genetic, like many other diseases, but despite decades of genome-wide association studies, today there is still no gene identified for depression or schizophrenia. So this has been always a very intriguing question, and also in terms of how to treat, how to perceive the patients and how to treat them. It has put a lot of difficulty, the fact that the etiology of the diseases was not known. But it's known that traumatic events, violence, abuse, abandonment or neglect during childhood or adolescence are responsible or are at least induce or favor the development of very severe diseases like depression, antisocial behaviors, cognitive and personality disorders, schizophrenia, addiction, and sometimes even suicide, and that many of these diseases run in families. And that it's not really through a purely genetic, it's not purely Mendelian, and again there has been no gene identified for most of these big diseases. So with this in mind, and I've always been interested I'm a molecular biologist as well, so I wanted to address this question from a purely molecular perspective. So we developed a mouse model many years ago, about 15 years ago now, to mimic this type of traumatic experience in early life. And for this we used a model. We tried different things, but the things which worked the best was unpredictable maternal separation. So it's separating pups from their mother every day, unpredictably, at any time during the day. for 3 hours per day. So it's a chronic manipulation, and from birth to 2 weeks after birth. And during separation, we also subject the mother to traumatic stress by putting her into different stressors like restraint stress or forced swing unpredictably. This combination of manipulation and the unpredictability of these manipulations are very important factors to determine the strength of these and the consequences of the manipulation. Here, we were interested in inducing symptoms which were transmitted across multiple generations. And this unpredictability was a very important factor into the induction of the transmission across generations. And here to generate offspring, so we subject the first generation F1 to this manipulation, and then we let them grow adult, we test the behavior, then we take the males, we breed them to wild-type, naive, non-traumatized females to generate F2 that we let grow adults, we test the behavior, and again we take the F2 males that we breed to females to generate F3. So here, F3, it's very important to go down to this generation because it's the first generation in which not a single cell, not even the germ cell from which these animals come from, has been exposed to the trauma itself. I'm showing here the Patry line, but we also did the experiment in the Matry line, and many of the data I'll show you are also valid through mothers, so there is also transmission through mothers. And here these females, they are naive, and we always use different females across generations, so to make sure that they have not been exposed at all. And the males, during breeding here, they are placed with a female just for a couple of days. They never see the offspring, so they cannot really influence the offspring. We also did cross-fostering at some point to really exclude maternal effects. So I'll show you today a few of the behaviors that we have examined and that we found to be transmitted across generations. In the literature, being exposed to traumatic event in early life often result into depressive behaviors, bipolar depression, for instance. In mice, it's quite easy to test depressive-like behaviors or behavioral despair. It consists in placing the mouse into a beaker of cold water. It's called the Posselt for-swim task, which was developed in the 70s by Mr. Posselt. And typically a control mouse will swim and try to escape. A depressed mouse will not fight and will start floating. So we measure the latency to float and the time spent floating. And it's a pretty good measure of depressive behaviors. We observe that in our mice, across generations, the time spent floating, but also the latency to float, so the time spent floating was increased, and the latency to float, I won't show this, but was decreased. And this is really across generations. At some point, we also tested F4 from the Patry line, and F4 animals, both males and females, are also depressed. So there is a clear transmission of depressive-like phenotypes. We checked this using another task, which is the tail suspension task, which is based on the same principle, and we also observe depressive-like behaviors. These animals are also anhedonic, so they cannot enjoy pleasure as controls. It's also another symptom of depression in human. Anhedonia, which is amplified by stress, is, however, transmitted to the F2, but not further down. But the clear transmission of depressive-like phenotypes. Another type of behavior observed in humans who have been subject to trauma is antisocial behaviors, and it's quite easy to look at social abilities in mice. You place animals who don't know each other in the same cage. There is a partition here, so you measure the number of times they look at each other, they sniff at each other. The first day they will do a lot of these recognition behaviors, and day after day, recognition behaviors or exploratory behaviors will decrease because they know each other. In our mice, social recognition is decreased, so I'm just showing here a discrimination ratio. You see that this discrimination ratio here is below the baseline because it shows social recognition in control mice. In MSUS mice, so in traumatized mice and the offspring, there is no social recognition, so they are not really able to read social cues when it's related to a peer. Their olfactory functions are normal. We tried it with food, for instance, so it's purely the inability to behave properly socially. Cognitive behaviors are also impaired, in particular memory. I'm showing here a object recognition task, so it's very simple. The animals are first placed in the presence of 3 novel objects that they will explore, sniff, learn about, and after an interval, which can be a couple of hours or 1 day or several days, they are placed into the same box with 2 of the familiar objects and 1 novel object. They can perceive the novel object as novel only if they remember the familiar object. And if they remember the familiar object, they will hardly explore them. They will spend most of the time exploring the novel object. And that's what we see here in control mice. So that's the exploration of the novel object during testing. You see that they do a lot. But the MSUS mice and their progeny, they can't distinguish familiar from novel objects. So they have impaired memory. That's object memory. They also have impaired phi memory. We tested different types of memory. So it seems to be a quite generic form of cognitive impairment across generations. So besides behavior, it's known in the human literature that stress is often associated with metabolic alteration. And this is also the case in our mice. We tested insulin and glucose in these mice across generations, while in the F1 there is no significant effect. There is a decreased level of glucose and decreased level of insulin, in our traumatized animals, F2, and decreased level of glucose. And you see that the glucose here at baseline and the response, the mounting of the glucose response after stress. And you see that there is a normal mounting of glucose response, but it always remains lower. So it suggests a kind of insulin insensitivity in these animals. We believe that the F1 animals are less affected than the F2 because here the trauma happens only postnatal, while in F2 all the machinery, as a consequence of trauma is present from the beginning, from the germ cell on. Now, what are the potential mechanisms? Because that's really what we are interested in. We developed this model, used behavior as a tool to demonstrate that there was transmission of behavior and metabolic functions, but the purpose of doing all this was really to be able to examine and identify the potential mechanism involved. And we looked at 3 possible mechanisms, 3 plausible mechanisms. So DNA methylation, histone, protamine, PTMs, and non-coding RNA, because I believe that probably all of these mechanisms are responsible, are contributing to some extent and differently. So I think it's important to really look at them in parallel. So DNA methylation, I will show you data on DNA methylation and non-coding RNA because this is still in the making. So but basically, this is a scheme of a sperm cell. And these mechanisms are plausible because DNA is known to be methylated in sperm cells, in adult sperm cells. Histones are known to be modified, so PTMs have been identified by mass spectrometry. However, in adult sperm, there are very few histones. In mice, it's between 1 and 5%. Histones are replaced by protamines during the later stage of sperm formation because protamines are much smaller and in nucleosomes, you need an octamer, you need 8 molecules of histones. In sperm protamine, sperm DNA, you need only 1 protamine. So there is no nucleosome, it forms a form of a type of tauroid DNA. But protamines are thus very abundant in the sperm chromatin, and up to recently it was not known if protamines can be modified or not. We looked at this by mass spec and we published a paper recently identifying a few post-translational modifications on histone protamines in adult males, and we found that they can be methylated, acetylated, and phosphorylated. But we don't know if these PTMs are altered by trauma. This is something that we are looking at right now. And non-coding RNA, it's known, as we have heard already, that small but also long non-coding RNAs are quite abundant indeed in adult sperm. So regarding DNA methylation, we used a very systematic approach and initially selected a few candidate genes which made sense for the behavior we were observing. Like here we have a receptor, so it's CRF receptor 2. It's one of the two receptors for the stress hormone CRF, or stress factor. And we used pyrosequencing, bisulfite pyrosequencing. So here it's just a scheme of the gene, and we did, that was almost 10 years ago. So today that may look a bit restricted, but we looked at the CPG island and we measured quantitatively DNA methylation by pyrosequencing in the whole CPG island, which is quite short, a little over 400 base pair. And what we observed was an hypomethylation in the sperm of the traumatized males. You see that there are several CPG, each bar is a CPG, and it's the methylation differential, so the difference between control and MSUS. And you see that there is hypomethylation in exactly the same stretch in the brain of the offspring, as well as in the sperm of the offspring. Here, the data is presented differently because we did this a couple of years later. But instead of methylation differential, here it's really the percent of methylation. And you see that the methylation is a few percent in the controls, and that it is decreased in MSUS sperm at different CPGs. And this correlated with an alteration of the expression of CRF receptor 2. We checked that there was a direct correlation by using 5-ASA, which is a GNMT inhibitor. So we took brain extracts, incubated it with 5-ASA, and looked at the level of methylation of CRF receptor 2 gene and the level of expression. And there was a correlation between the level of DNA methylation and expression. So it's only correlative, but that's the best we could do. So you may think here trauma may have altered the epigenetic machinery or the DNA methylation machinery, and there will be probably many other genes which are hypomethylated. There are other genes which are hypomethylated, but it's the alteration in DNA methylation are indeed bidirectional because there are genes, many genes, which are hypermethylated. And that was the case for MECP2. MECP2 it's a gene which is involved in Rett syndrome, a severe neurological disease. It's a methylated DNA binding protein. And like for CRF receptor 2, we did it quite systematically. We measured the level of DNA methylation by bisulfite pyrosequencing in the CPG islands. And here we found that this stretch, several CPGs in this stretch here, strategically positioned right before the ATG, were hypermethylated. Not only in the sperm of the traumatized males, but also in the brain of the offspring and in the sperm of the progeny. And that this correlated with a decrease in the level of expression in the brain. So which again suggests that DNA methylation is altered in different tissues and across generations, at least down to F2. Some genes are not altered at all. So this is what we have for DNA methylation. Now I'd like to turn to non-coding RNAs. So we initially approached the question also in a systematic fashion by doing deep sequencing on the sperm from the traumatized males. So we had 3 samples, 3 control samples and 3 MSUS samples, and each sample here is the pool of the sperm from 5 males. So the ends here are quite large. And you have here the list of the microRNAs or the small non-coding RNAs which are affected and the fold change. And the code color is blue is low level of expression, yellow a high level of expression. And you immediately see that in the MSUS samples there is increased expression. There is a lot more yellow, in particular in this sample here. So overall, a overexpression or an excess in some microRNAs in the sperm of traumatized males. We confirmed this by qPCR and focused on a few microRNAs, those which were found to be the most overexpressed by deep sequencing. So this is here microRNA expression in F1 sperm. You see that it's highly significant. And again, like DNA methylation, these changes were not found only in sperm, they were found in the serum. While this one, this microRNA was also overexpressed, the others were downregulated. But it was also, the microRNAs were also overexpressed, at least some of them, in the hippocampus, so in the adult brain of the same males. And this was also transmitted to some extent to the offspring. Some microRNAs were downregulated in F2 serum, and many more than in F1 actually were overexpressed in F2 hippocampus. So again, a form of epigenetic alteration, which is across different tissues and across at least 2 generations. There was no change in the level of microRNAs in F2 sperm, although the phenotypes are transferred to F3. So we don't know why this is the case, but we believe that maybe the microRNAs are the initial trigger, and that they may be the memory or the signal may be transferred to another form of epigenetic factor, epigenetic mark. We don't know. Now, to provide the causal evidence for DNA methylation, that's very complicated and at all not possible, I think, because in our case we have hypomethylation on some loci, hypermethylation on others, and indeed we looked only at the CpG island, so it could be that in a single gene there is hypomethylation of the CpG island but hypermethylation of another fragment of the gene. And to mimic this artificially, even with CRISPR-Cas9, it's not possible because it involves too many loci and opposite changes. However, for the RNA, now that you have heard François talk about the possibility to inject RNA into fertilized oocytes, I think for us it was a perfect type of strategy, and inspired by the work of Minou. Indeed, that's what we did. We extracted the RNA from the sperm of traumatized males, and we injected, indeed that was the same total RNA as the one we use for RNA sequencing, and we injected it into fertilized eggs from control mice. And after injection, we transferred the injected eggs into pseudo-pregnant females by surgery, and then waited until the progeny was born. So these are the animals arising from sperm RNA injected eggs. We tested the behavior and we bred them to wild-type females. We use the males to generate the progeny. And we could reproduce some of the phenotypes by this approach. You see the performance on the 4-swim task. They spend more time floating, so indicative of a depressive type of behavior. Here there is a decrease, it's risk-taking. I did not show this data, but our animals also have more, they have less risk assessment behaviors, and they take a lot more risk if they have, for instance, the option between a foreign, potentially dangerous territory and a very safe territory. They will prefer the dangerous territory. And that's shown on this task here. So a phenotype that we could see systematically in MSUS mice was reproduced here. So this is now in these animals directly arising from the RNA-injected eggs, but that was also the case in their own progeny. So their own progeny was also depressive. So here it's a pretty good causal evidence between the RNA, the sperm RNA extracted from these traumatized males and these phenotypes. Not all phenotypes were reproduced. We know in these mice, for instance, that synaptic plasticity, long-term potentiation, for instance, in the hippocampus, is deeply, it's dramatically altered. It's not possible to induce LTP. In these animals here, LTP is normal. So which suggests that the RNA can carry some of the effects of trauma, but not all of the effects. At the moment, we have recently developed artificial insemination to test the possibility that to transfer only sperm to exclude all other factors. We are very convinced that there are other factors involved, but it's one of the last strategy that we would like to use to really have a final evidence that it's through non-maternal or non-paternal effects. So yes, indeed, in addition to the behavior, the decrease in insulin and the decrease in glucose were also reproduced in the animals arising from the sperm RNA injected eggs. So I'd like to stop here. So I showed you that traumatic stress in early postnatal life can induce depressive behaviors, antisocial behaviors, and memory deficits. For those of you who may want to know whether all the behaviors are negative or are not advantageous. No, that's not true. We tested the idea that perhaps in challenging conditions, being exposed to trauma could be positive. And indeed, in these mice, when they are exposed to a condition where they are very thirsty, they need to learn the rule of a complicated task where they have to learn an interval between a signal and a lever pressure that they have to press to get reward. They are better. They are better when the rules change. If the interval is increased without them knowing it, they can adapt faster. They are more efficient. They are able to use strategies in challenging conditions that control mice take much longer to use. So in terms of evolution, I think it's a very important aspect to say that being exposed to stress in early life can make individuals maybe more responsive in some conditions, which is true in human. I showed you that the behavioral symptoms are transmitted to following generations down to F3 through males. We tested transmission through females down to F2, and this is also the case. After cross-fostering also. Traumatic stress alters the profile of DNA methylation bidirectionally in the brain and male germ cells in sperm, and impairs gene expression. And but besides DNA methylation, we believe that there are other non-genetic factors such as sperm RNAs which are involved in the expression and in the inheritance of some of the effects of trauma. And this is just a picture of my group at a retreat by a Swiss lake. And I'd like to thank really the multiple students, postdocs, and trainees who contributed to this project, and the generous universities and funding bodies who are supporting us. And I thank you for your attention.
Merci. Question Michel Ladzinski.
Thank you very much, Isabelle, for this very, very nice talk. An experiment that I would like to see is the reverse experiment. So it's even more important for humans to know. So you subject your mice to a variety of stresses. They are then affected in post-traumatic stress, in depression and social behaviors. Now, these mice at the second or third generation, you put them in a very positive environment. What happens?
Yeah, we did this experiment actually, and we just submitted the study. We took F2 adults, young adults, 2 to 3 months, and we placed them for several weeks in an enriched environment. So there are large cages where they are in social groups. There are toys. There is a maze that they have to cross. Yeah, no, but we could for 3 weeks, and then we test their behavior. We tested the behavior before they were placed in these enriched environments to make sure that they were expressing the phenotype, and after. And for depressive behaviors, you can do this with the tail suspension task, for instance. And being exposed to this environment and enrichment reversed the phenotype. So they were no longer depressed after this exposure. Now, we also looked at DNA methylation in the sperm to see whether it was reversed. And for some genes, at least, we could see that there was a correction of the epimutations on some genes. We also bred these males, generated the following offspring, and the offspring, after the males were enriched, the offspring was not depressed. So it's only, it's a focused study on very specific behaviors and a few genes that we checked. I'm not making a major statement here that everything is reversible. I don't know. I don't know, for instance, if the RNA alterations are reversed. This we have not tested yet. But at least some of the phenotypes which can be correlated with epimutations, with some alterations in DNA methylation, can be corrected.
It's important to present the two faces for giving hope.
Sure. Yeah. Yes. Well,
and if you treat the animals with, for example, fluoxetine, simply to decrease the depression, you treat them for some time with fluoxetine at third generation. They would be good in a poor salt assay or other type of assays.
Yeah, we did this.
What happens?
We did this
the
not
next generation.
Yeah, we did this not with fluoxetine, but with imipramine.
Okay.
But that was 10 years ago when we were mostly interested in the reversal of the behavior itself.
Yeah.
We did not breed these males after treatment, so we don't know. But now it's known that some antidepressants dysregulate non-coding RNA in blood, at least. And I'm not sure if it's been done in sperm, but it's very possible.
It's important to understand the mechanism for humans.
Yeah, absolutely, absolutely.
Vincent Collot.
If sperm RNA is so important in shaping the fate of the progeny, can one imagine now systematically doing experiments in which you make, instead of doing hybrid crosses between two strains of mice, you take your inbred mice, but you systematically now inject RNA from sperms from outbred mice and see whether, to which extent first you get phenotypes, how persistent they are, and therefore get a sense of how relevant, sorry, not how relevant, but how penetrant or how comprehensive these phenotypic alterations governed by RNAs might be at a species level.
Sure. This may apply again to some phenotypes or some traits, not all.
Yeah, but how does one choose phenotypes? You're interested in neural phenotypes and you found what you wanted. But my question is more general one. So how much this kind of inheritance mediated by small RNAs carried by sperm affect indeed the phenotypes?
Yeah. I'm not claiming that the inheritance is due to small RNAs. We don't know. It's total RNA. So we...
No, I'm not, sorry. RNAs.
Yes, RNA. It's very important
RNAs.
Because some people may think that it's microRNA.
I may have said small RNAs, but that's a mistake. RNAs.
Yeah. We are indeed doing the experiment right now. We have separated the population of small RNA and long RNA, repeated the injection to see whether we can distinguish the populations. But to answer your question, yes, I do think that there are non-coding RNAs, but messenger RNAs in sperm could also be involved, which are altered by a mechanism that we don't know. Maybe it's through DNA methylation of the promoter, we don't know, which are altered and which can keep a memory of an experience, whether it's a traumatic experience or a diet or any kind of experience, and that this memory can be maintained and transferred to another body.
Claudine Julien.
It is, although it is, it is controversial still. Depression seems to be different in women and men. So, do you have in your experiments differences between males and females in terms of depression?
Yes, indeed, that was shown in some of the data. It's true that in a given group, siblings, males may be depressed and their sisters not. In one generation, and in the following generation, you may have a male which is asymptomatic. It won't show obvious signs of depression, but still the progeny will be depressed. It's something we don't understand. It's something when you talk to psychiatrists, it's very common in human that there is a kind of the possibility for men or for women to carry some factors which make their progeny depressed, but themselves they don't express it. But we do see differences, yes. In behavior, there are many behaviors which are subjected to sex differences.
So does it mean that they probably would need a challenge to express their susceptibility to depression?
Possibly the tasks that we are using are not appropriate, and maybe there is this theory of the two-hit model that you have a kind of a susceptibility, and when you are exposed to a challenging situation, then it's only then that the behavior shows.
Yeah, so they may be different to this
Absolutely.
challenge.
Yeah, we have not gone so far in our analysis, but it's a good point.
We have this in mice that are resistant to the high-fat diet, and we can see that from the same mother. And therefore your animals can express or not the susceptibility.
Yes.
Thank you.
Two more questions. Eva Jablanca and then François Cousin.
Yes, I wanted to ask you what you think is how depression affects this. Do you think that what is happening is a change in the neurohormonal system that is affecting in parallel the brain and the sperm. So this would be a case of parallel induction. Or do you think that it is an effect on the brain, which then is transmitted somehow, let's say through exosomes carrying small RNAs or whatever, to the, from the soma to the germline? I think in theory it would be possible to distinguish between these two possibilities.
It could be both. The first option is that so you have a stress which modifies something in the circulation, let's say, and which affects all organs. So to test this idea, what we did recently was to extract serum from traumatized pups at P7 after 7 days of trauma or after 14 days of trauma, and do a high throughput proteomic and metabolomic analysis with no theory behind, just high throughput. And what we observe is very interesting. Well, there are many targets that we don't understand, we don't know what they do, but there are many proteomic and metabolomic alterations. In the metabolomic, metabolic factors which are altered, some are known to be able to bind to receptors at the surface of spermatogonia, for instance. And we are now in the process of trying to test this idea. So the first thing to do is to culture or to have spermatogonia in culture, put these metabolic agents and see whether DNA methylation in some targets is altered. But it's very speculative, you know, we are really, at the moment we are faced with these dozens of pages of compounds which are altered in a way, but we don't know how. We will have to link it to the epigenetic machinery. It does not exclude the fact that perhaps there are exosomes which are released from the brain, early development, since the blood-brain barrier is not completely closed. The blood-testis barrier is also not completely closed, so it's possible that there are exosomes which are transferred there, but that would be, I think, more difficult to prove, perhaps by labeling exosomes in a way, or identifying some microRNAs and seeing whether they are transported, but yeah. But that's a very plausible mechanism as well.
Last question by Isabelle, do you think the change in methylation of the promoter that you see could be by itself also a transgenerational signal? It would have to escape the wave of demethylation and remethylation in the embryo. Some idea about that?
Yeah, I think it does, since we see it in F1 and F2 sperm. We did not systematically look at F3 or F4 sperm, but at least I think there is a chance that at least some of the fragment of the genes of interest escape reprogramming. Whether it's erased and re-established for the excess in DNA methylation, or whether it's really maintained, we don't know.
Talking about your talk. Thank you very much.